Photosynthesis in plants. So photosynthesis is an endogenic process. It means it uses energy. It is an anabolic pathway. It means a synthesis of complex compounds. Oxidation to reduction process converts solar energy into chemical energy. Equation of photosynthesis is 6 CO2 plus 12 H2O in the presence of chlorophyll and sunlight gives C6H12O6 plus 6 H2O plus 6 CO2. Site of photosynthesis is chloroplast. There are few experiments in this chapter. First is mole's leaf experiment. So this is a plant, and this plant, this leaf, CO2, to prove that it absorbs CO2, this leaf is taken out, and it is half exposed and half is unexposed to CO2. It is half is covered by black paper or something. So this half is which is exposed gives positive starch test, and which is unexposed to CO2 gives negative starch test. So negative starch test or positive starch test is an iodine test which gives blue black color when dropped iodine in that leaf. Now next is Joseph Priestley experiment. It is Joseph Priestley is awarded for discovery of oxygen in 1774. So this is a candle and this is a mouse. In next and this whole setup is covered by a lid. So when we covered by the lid, there is no oxygen, and this mice releases carbon dioxide, so it dies. Now in the next step, this whole setup is same, but one plant is added. So the whole thing is this, and the candle is also continuous burning. In this candle does not continue burning. So this candle releases foul air as well as this mouse. So this is foul air which is carbon dioxide, and this plant releases good air which now we call as oxygen. It is a mint plant which is sustainable. This whole setup is sustainable because of this mint plant, because this mint plant releases good air. So this good air and this this good air is called as oxygen, and this foul air is called as CO2, which is released by respiration of rat and burning of candle. So this proves the importance of oxygen, involvement of oxygen in the the photosynthesis, and this mole leaf experiment proves the importance of carbon dioxide in the process of photosynthesis. Next is Jan Egonhaus experiment. It proves sunlight is important for photosynthesis. So there are two setups. First, in which hydra is in both hydrilla is a plant. So in first setup, hydrilla releases bubbles in which proves O2 oxygen is released, and another hydrilla no bubbles are produced. It is because the first setup was provided with the sunlight, where the next setup was not provided with the sunlight. Thus, no oxygen is produced. or released, which means no photosynthesis is performed. Hydrolyzed submerged hydrophyte. Next is Julius von Sachs experiment. He concluded that simple compounds stored in the chamber that perform photosynthesis. Now simple compounds here now we call as chlorophylls, and special chamber is called as chloroplast. Engelmann experiment. In this, it is a very important experiment which often comes. So, a prism is taken. In this prism, side of this prism, this is a glass tube in which cladophora is kept. Cladophora is a green algae and is an aerobic bacteria which utilizes the oxygen. So cladophora is kept, and for photosynthesis blue and red wavelength of light is used. So now that's why I have shown only red and blue light. So this is the light rays. It passes through the prism, and here we get all the 7 colors of light. Sorry, here we get red light and blue light. This shows that for photosynthesis only blue and red wavelength of light is used. Next, sixth experiment is actually it is a conclusion by Van Niel which shows that before 1930 the equation of photosynthesis was CO2 plus H2O plus CO2 plus H2O plus CH2O plus oxygen. H2S is used by sulfur bacteria, produces sulfur gas. So H2O, which is used in the photosynthesis, should release oxygen. Next experiment is Rubin-Kamen experiment, in which O18 which is a non-radioactive isotope is used, in which H2 used for experiment, oxygen was collected back. So this H2O in which oxygen is O18. So after the complete process, oxygen which was released was collected back. So it was concluded that it was O18. This proves that oxygen which is released in the end process is formed from water, which comes from water. So the new equation is 6CO2 plus 12H2O in the presence of chlorophyll and sunlight gives 6C6H12O6, which is glucose, plus 6 oxygen plus 6 H2O. Next is site of photosynthesis. Chloroplast is a site of photosynthesis. This is a chloroplast cell. This is the outer membrane. This is the inner membrane. And it is intermembrane space. This is thylakoid lumen. This is whole thing is thylakoid lumen, and this is grana, or plural is grana, singular is granum, and inside is thylakoid lumen. This is circular double-stranded DNA and 70S ribosome. That was this chloroplast is also called as semi-autonomous organism, semi-autonomous organelle. This is stroma lamellae. Stroma lamellae is site of light reaction, which is a cyclic process, and thylakoid is a site of light reaction, and thylakoid is a photophosphorylation, non-cyclic photophosphorylation, in stroma, site of dark reaction. Pigments used which harvest the light: chlorophyll A, chlorophyll B, xanthophylls, and carotene. Chlorophyll A is a greenish blue in color. Chlorophyll B is olive green in color. Xanthophyll is yellow in color and carotene is orange red in color. This is increase in the increasing order of molar weight. So orange red is less than yellow is less than olive green is less than greenish blue. Now one main pigment, that is first is main pigment, is chlorophyll A is the main pigment. Now 2, 3, 4, that is shield or accessory pigments, which is for protection of chlorophyll and help in harvesting of different wavelength of light. That is because blue and red are used in the process of photosynthesis, so other pigments help in the harvesting of other wavelengths of light. Light also increase in the absorption spectrum. Chlorophyll A soluble in petroleum ether. Chlorophyll B soluble in 92% methyl alcohol. Now xanthophyll is soluble in ethyl alcohol, and carotene is soluble in petroleum ether. Now photosystem. So this is reaction. This is a complete photosystem in which this is photosystem. This topmost point is containing chlorophyll, which is reaction center, and the bottom part is light harvesting complex or LHC or antenna, which contains accessory or shield pigment. It is chlorophyll B, xanthophylls, and carotene. Now this has two parts. First is PS1, that is photosystem 1, which has also chlorophyll and which harvests the wavelength of 700 nanometer. And another is photosystem 2, which is PS2, which harvests the light of 680 nanometer. Name is on the basis of evolution, not on the basis of function. Now structure of chlorophyll. It is a tadpole-like structure. This is middle part is Mg. This is porphyrin head, tetrapyrrolic, and four pyrrole rings, and phytol tail. Anchor of chlorophyll to thylakoid membrane. Molecular formula is of chlorophyll A is C55H72O5 N4Mg. Light reaction. Now light reaction takes place in the presence of light in thylakoid membrane. Light reaction takes place in the presence of light. There are two types of light reaction. First is non-cyclic photophosphorylation, another is cyclic photophosphorylation. Non-cyclic photophosphorylation site is thylakoid membrane oppressed. Requirements are PS1, that is photosystem 1, PS2, photosystem 2, ATP synthase, NADP reductase. So for non-cyclic photophosphorylation, we require PS1, PS2, ATP synthase, and NADP reductase. For cyclic photophosphorylation, we require PS1 and ATP synthase only, and stroma lamellae is the site of cyclic photophosphorylation, which is unoppressed. Now later we will discuss what is oppressed and what is unoppressed. Now this whole thing is called as non-cyclic photophosphorylation, also called as Z-scheme or Mendel or Hill reaction. So first is it is electron. First this light falls on PS2 of 680 nanometer. It falls on PS2 on the reaction center. H2 is used, and it's converted into 2H plus and 2 electrons, half of oxygen. Mn2 positive, Ca2 positive and Cl minus is used in this process. Now this half reaction releases by the diffusion, and this electron is transported here in uphill. This is an uphill reaction. This again, this is the same electron with the help of pheophytin. It is a downhill reaction, transports to phastoquinone, which again transports to cyto B67 complex. This transports to plastocyanin, which is 2 Cu centers. In center it is copper. It is according to increase in redox potential, it is a downhill reaction, while this is an uphill reaction. Now this plastocyanin gives this electron to PS1, and this PS1 takes the light of 700 nanometer, releases this 2 electron, and this 2 electron is given to FES protein. FES protein gives it to ferredoxin FES, and this FRX complex converts NADP plus H plus to NADPH2, and this H plus comes from stroma lamellae. Okay, this is lumen, and this whole thing is the whole electrons are coming inside the lumen for synthesis of ATP by chemoosmosis. Now here we used a complex for in PS2. This is called as oxygen evolving complex. In PS2, Mn2 positive, Ca2 positive and Cl minus is used. Products are O2, ATP, NADPH2 assimilatory. ATP and NADPH2 are assimilatory power. FRX, it is ferredoxin redox substance, which we used here. One electron accepted from PS2 is 1 degree electron accepted from PS2 is pheophytin. 1 electron accepted from PS1 is FES protein. Final electron accepted from PS2 is PS1, and final electron acceptor of PS1 is NADP plus. H2 is external electron donor. So this H plus comes from H2O.